Good morning, everybody. Donna, sorry, Dana, can you hear me okay?
Sure can.
All right, great. Good morning, folks. This is Mehul Mehta. I'm one of the three co-chairs for the session, in addition to Jim and Paul. I want to welcome you back to the workshop. As soon as the slide is removed, we are on break, but I think in a minute or so it'll be removed. We are in the second session now, and I'll be introducing the next four speakers. After that, we'll move to the panel discussion immediately after that. Then we have the break, and after the break, we'll have Jenny Trestman present her views on future of permeability, and then we have the breakout session. So let me start with introducing our first speaker for this session, Dr. Ravi Shankar. Ravi is a senior research fellow in drug product design with pharmaceutical sciences at Pfizer worldwide research and development in Groton, Connecticut. Ravi has over 32 years of research experience in innovation for several new novel drug delivery technologies, advanced mechanistic approaches for drug discovery support and industry, leading experimental as well as computational biopharmaceutics. So a wealth of experience from Ravi Shankar, who will be sharing his viewpoints on BCS aspects on the innovator side from the new drug application point of view. So Dana, we can have Ravi's presentation now. Thank you.
Hi, this morning I'm going to share with you the successful application of BCS-based biowaivers in NDAs. I'd like to mention at the outstart that we have recently received requests from the FDA for several of our recent NDA filings to provide such biowaiver justifications. So we at Pfizer have internally decided to strategically provide this type of justification in future filings. The outline of my presentation is as follows. I'll start with an acknowledgement, which will include a disclaimer. Then I'm going to highlight the two enigmas that are faced by the pharmaceutical industry and how those enigmas actually then relate to the BCS categorization with a focus on permeability, and also how these permeability uncertainties manifest itself into the uncertainties associated with the BCS categorization and the implications that has during the development of the product. I'll also give some examples of biowaivers, of how the BCS-based biowaiver has been utilized to support different products, and provide information about future directions, and then conclude my presentation. First of all, I'd like to thank the organizers of this workshop for giving me an opportunity to share my experiences. I'd like to also take the opportunity to thank numerous colleagues at Pfizer who have greatly enriched my knowledge by sharing their knowledge with me and their experiences with me, and that has enabled me to give a short presentation here today. Additionally, each drug molecule that we have advanced from discovery into clinical development has provided unique opportunities to learn, and also it has increased the level of humility that I have towards data. Gaps in fundamental science continue to present significant challenges, especially as it relates to oral absorption and the BCS categorization itself, and making broad generalizations for biowaivers. At the moment, and probably in the near future, each molecule will continue to require its own unique justification for biowaiver. I do have a disclaimer that the views that are being expressed here today are my personal views, and they are not the views of Pfizer. I would like to point out two predictability enigmas that are faced by the pharmaceutical industry. And these two enigmas are the ability to accurately predict solubility and permeability, and not just accurately predict, but also with confidence from molecular structure. The lack of confidence in predictability are due to the gaps in basic science. And the gaps in that science is really provides a disconnect between theory and experimental observations. So it's interesting that these two parameters that we cannot readily predict forms the basis for the BCS classification itself. So despite these enigmas, it's a profound interest for all of us, especially the industrial scientists, to convince regulatory agencies worldwide to grant biowaivers for some drugs based on the evidence and convincing scientific aspects of that evidence. So it's a significant challenge, but it also makes it possible for us to have job security, both in the industry and academia, as well as probably the regulatory agencies as well. Although the prediction of solubility and permeability, which are the two parameters in the BCS categorization, continue to be the holy grail in science, there are experimental techniques for both that are utilized quite extensively to enable the BCS categorization of new drug molecules. One of the questions that really does come up is that how relevant are these measurements relative to the human physiology, and what is the confidence of these estimations in the human physiological fluids? There's good guidance from regulatory agencies from different parts of the world for determination of solubility, and there have been further advances that have been occurring about how to subcategorize the solubility, say for example BCS class 2A and 2B drugs, and incorporation of bile salt mixed micelles in terms of determining the solubility. These are available readily for scientists both in academia and in industry. However, the quantification of permeability inherently presents significant challenges throughout the drug development process. It's not only the measurement, but the interpretation gets confounded due to changes in flux resulting from the solubilization of drug molecules due to either physiological or formulation factors. Biopharmaceutics scientists know extremely well that solubility and permeability in the gastrointestinal tract changes by position. Although pH solubility considerations are easily understood and they can be measured in vitro quite readily, the regional differences in permeability arising from physiological and anatomical differences are much more difficult to get into the BCS categorization. This leads to a higher degree of uncertainty in the assessment of permeability during the drug development process. Most biowaiver considerations based on regulatory guidance documents are inherently for BCS class 1 and class 3 drugs. Since both BCS class 1 and 3 drugs have high solubility, does it imply that permeability has inherently low importance in the justification of biowaivers? And thus the uncertainties associated with permeability is not as big an issue. I guess that is not the case because this entire workshop is focused on the importance of permeability. So the permeability assessment during drug development presents significant challenges because of the techniques that we may be utilizing during measurements at the various stages of development. During early stages of compound design and selection, we typically use high throughput cell-based permeability assays. And the choice of cell lines are many. Each company has its own preference, and there are standardized methods that are utilized internally within a company about what concentrations to use, what pHs to use, what number of passage of cells, the reference standards, the incubation time, etc. However, when the compound comes into phase 1 clinical studies, there might be additional investigations that might be conducted to understand the permeability of the molecule, especially for ionizable compounds. Historically, at Pfizer, we used to determine rat single-pass intestinal perfusion studies to assess the permeability of the compound and then do the correction factor for estimating the permeability in humans. However, this has not been a routine best practice for the past 10 years. ADME studies provide a significant amount of information by the utilization of radiolabel studies with mass balance studies, and nowadays with microdose studies to get a good estimate of the fraction dose absorbed, and as a result, a good estimate of permeability. However, something that we face constantly in the industry is that during drug development, most of our molecules come into the category of intermediate permeability. Although BCS requires us to define the permeability to be either high or low, but the behavior of the compound and the measurements that we make lead us to believe that the compounds quite often have intermediate solubility. So how do we reconcile to all of these pieces of information? The uncertainties in this BCS categorization that arises from the difficulty of making a good assessment of permeability continues to be a significant problem. And there are confounding factors, as I mentioned before, especially due to the solubilization from physiological and formulation factors. The assessment of fraction dose absorbed has helped quite a bit, but that also becomes challenging when molecules are subjected to either intestinal first pass absorption or the absorption is influenced by transporters, whether they are uptake or efflux. So uncertainties in BCS categorization is a significant factor that makes justification for biowaiver a significant challenge. In addition, permeability is not the only factor. There's a significant amount of dissolution testing that needs to be conducted, especially utilizing multi-pH media to provide justification for a biowaiver. And in some instances, depending upon the solubility of the molecule, it may require the use of agents like surfactants to demonstrate complete and potentially rapid dissolution of these molecules. The interpretation of all of that again presents some significant challenges. This slide is a summary of the different types of biowaivers that we have had direct experiences at Pfizer. And I call it that these are partially successful applications. And that will become very clear in a moment. For BCS class 1 drugs, especially new drug application, there are limited number of examples. But when we have applied those and utilize biowaiver for low dose strength tablets, utilizing common blends for making the different dose strengths, this has been relatively well accepted in majority of the markets. When we come to BCS class 2 and class 3 drugs, where we have conducted RBA studies to demonstrate equivalence for API with different particle size, with different formulation, different processing conditions, the biowaiver for either new dose strengths or different dose strengths with common blend, that has typically been accepted only by the FDA or maybe EU. So for worldwide filings, we end up conducting BE studies. When you get to BCS class 1 and 3 drugs for legacy compounds, which we have a large number of molecules, because Pfizer has had drugs on the market for 50 plus years. And when we go through SUPAC changes, whether it's a site change or a process change or some formulation, composition changes, or there might be some minor changes, we may conduct all of these investigations where BCS-based biowaiver could be justified, but we find that these are not accepted globally. When we go to BCS class 1, 2, and 3 drugs, where the dissolution at one of the pHs may be behaving very differently, or there's incomplete dissolution, then the ability to get biowaiver reduces significantly, and especially the global acceptance of biowaiver continues to be a significant challenge. So BCS biowaiver from a global acceptance perspective is a significant challenge overall for us. And Pfizer, like many major pharmaceutical companies, we want our products to be registered globally. So if we do not get biowaivers globally, and we have to conduct BE studies for global registration, then the entire basis for submitting biowaiver starts becoming less lucrative for us. What we want to see is what is the impact of ICH M9 guidelines and how that will actually change the landscape is yet to be seen. The BCS-based biowaivers for global filing has not been very promising. The justification for biowaivers for legacy products has also not been universally been accepted. In addition, for any biowaiver that is based on permeability consideration is actually not the key driver, but the dissolution data is often the key driver for the biowaiver justification. So what we see that these challenges actually make us think about, is there a better option in the future? And one of the future options that we do consider quite significantly is the implementation and utilization of PBPK or PBBM type of modeling. But the challenge still is that even if we are able to verify and validate such models, would they be accepted globally? We could probably utilize PBPK modeling to demonstrate sensitivity to parameters such as permeability and or dissolution. But would it still be accepted globally? We may go to the extent of utilizing PBPK modeling to provide clinically relevant dissolution specifications. However, despite these efforts, would these be accepted globally? As I mentioned already, the outcomes of ICH M9 is still very unclear. We would probably want to see in the future improvements in quantitative assessment of permeability of molecules, if that could help us with biowaivers. So here are some key conclusions from my presentation, as well as our own learnings at Pfizer. With the body of evidence that we have to date, that BCS-based biowaivers in NDAs is not extremely common for the perspective of global filing. The application of BCS-based biowaivers for legacy products is also not universally accepted, even for BCS class 1 drugs, which is very unfortunate. The amount of dissolution data that we have to generate in support of a biowaiver is quite extensive, but there is no confidence that there would be a successful outcome for those biowaivers. Even with compounds that have high permeability, if dissolution data is not acceptable, even at 1 pH, or due to high variability at the first time point in the dissolution studies, then the probability of the grant of biowaiver becomes significantly reduced, especially by some board of health authorities worldwide. We have conducted, as a result of all of these outcomes, many BE studies, and we have rarely seen BE failures. So despite the body of evidence of BE successes, we have very limited amount of success in terms of biowaivers globally. So I end this presentation with a thank you for the opportunity that you have provided, and I thank you for your attention. Thank you.
Well, thank you, Ravi, for that very informative presentation from the innovator point of view and challenges faced by innovators for global submissions. Yeah, those are some real practical challenges, and we do hope that M9 implementation becomes more uniform across the globe and its implementation. Now I'm going to introduce the next speaker, which is Dr. Sid Bhoopathy. Sid is the Senior Vice President of Operations at Absorption Systems, which is a Pharmaron company. In this capacity, Sid's primary responsibility is to execute the scientific and strategic growth of Pharmaron Lab Services and CGT in the US. Sid is a real expert with a lot of experience in the area of conducting absorption studies, especially with KECO2, with Absorption Systems company, and been presenting those findings, at national, international meetings for a period of time. I'm really looking forward to this presentation where he's going to share industry case studies of successful permeability studies supporting BCS-based biowaivers on the generic drug side, ANDAs. So with that, if we can start Sid's presentation. Thank you.
Hi, good morning. I'd like to thank the organizers for the invite. Today I'll be speaking about successful permeability studies supporting BCS waiver in ANDA applications using the KECO2 test system. I will be sharing my thoughts and experience on best practices for successful permeability studies. In the case studies that I'll be describing, all the studies were successful except for my very last example, which is on a BCS3 product, which is still undergoing review and the outcome is pending. My presentation is divided into 4 categories of case studies. The first category is recovery-based questions. The second is case studies around permeability procedures. The third, I have case studies on test system or method suitability. And finally, I also have a case study on the impact of excipients. The format of the presentation is question and our response, question from the agency, and how we have responded to the question or approached the response to the question. So let me begin by recovery. A couple of definitions so that we're thinking about this the same. Solution recovery is the recovery from the donor and the receiver compartments. Mass balance is solution recovery plus recovery from cell lysate and a combination of donor and receiver rinses. Now with mass balance, there is typically an acceptance criteria expectation of more than 80%. And if this number is not met, that opens up potentially the instability box, which can be tricky to navigate. So as we prepare for the pivotal permeability study, we evaluate non-specific binding to a cell-free transfer device, and also we assess recovery from a cell-free transfer device where the components of the device, the top and the bottom, are evaluated separately. So as you can see from this example, non-specific binding to the combined device is about 50%. Individually, it is between 69 and 77. typically when we see recoveries below 85%, we start thinking of intervention measures. This could include pre-incubation of the drug substance before we begin the main permeability experiment. It could include collection of cell lysate and donor and receiver rinses to obtain mass balance, and also collecting receiver samples with organics so that we do not underestimate the concentration that has migrated across over the course of the incubation duration. So with that back, the first question within this case study of recovery, a 10-minute pre-incubation with test compound was performed to reduce non-specific binding. However, the concentration and volume of drug substance was not clearly mentioned. So this appears to be more procedural, not providing sufficient amount of details, not super difficult to respond to. So our response was the pre-incubation step was performed with the same concentration as in the main experiment. As in the main permeability experiment, we use the same dosing volumes and the dosing solution that we would in the main permeability experiment. After the 10-minute pre-incubations, all solutions were removed and replaced so that we have a clean baseline to initiate the permeability assay. The second question under recovery are our agency's expectations on recovery. So in this question, it is noticed that in the permeability study, recovery is higher when the concentration of dosing solutions, so recovery here refers to solution recovery, is higher when the concentration of dosing solution is 5 micromolar versus the recovery when the concentration is 50 micromolar. You have mentioned in your submission that the recovery would be higher as the concentration of test compound in the dosing solution increases. But however, this appears to go in the opposite direction. Can you please provide the reason or explanation on this discrepancy? So the increase, we typically observe increase in solution recovery with increasing dosing solution concentrations, but that was not the case in this situation. So the question is why? Our response, when we calculate recovery, it is typically based on the nominal dosing concentration, depending on the performance or the measured concentration of the dosing solution. But for compounds that are sticky, using nominal may create the appearance of atypical differences in solution recovery. So a better approach for sticky analytes, which could be where the dosing solution preparation is biased or you have differences in non-specific binding and cell accumulation, a better approach is to calculate recovery normalized to the initial available donor concentration in that insert or transfill. So when we approach, the results become much more typical or in the direction of what is expected, where the solution recovery at the higher concentration of 50 micromolar is greater than the solution recovery at 5 micromolar. And additionally, mass balance, which is the more important measure, where you're trying to show that you're able to recover everything that has been placed on the donor side, is complete at both of these concentrations, 99.4% and 102% respectively. The next question under recovery, also subcategory of recovery expectations. In the bidirectional permeability study, the average recoveries of drug substance from basolateral to apical direction were higher when compared to recoveries from apical to basolateral direction at multiple concentrations and in different studies. Please explain these differences in recoveries, solution recoveries between basolateral to apical and apical to basolateral directions. And this happens a lot when you have compounds that have certain physicochemical properties, high log Ps, high log Ds at the donor pH conditions, where you have issues with non-specific binding or association with the cell monolayer, a considerable fraction of the compound is recovered post-experiment in the cell lysate. Now, the differences in cell accumulation are largely attributable when you go A to B direction, the direct placement of the dosing solution on the apical surface of the cell monolayer. And that is not the case when you go in the B to A direction. So the morphology of the differentiated cacomonolayer on the apical side contains microvilli, which amplifies the surface area, and therefore there is a greater possibility of cell accumulation or cell association A to B versus B to A. And this indeed bears out, as you can see in the middle of the slide. There was greater accumulation of cell monolayer compartment apical to basolateral direction versus the B to A direction, 28 versus 8%. And this was essentially the reason why solution recovery is lower A to B versus B to A. The results in some more detail. This has solution recovery plus all the different compartments that were analyzed. In the A to B direction, solution recovery has a mean of 52%. Recovery from the cell lysate, 28% versus the table at the bottom, which is in the ballpark of 8%. And the rinses are very, very comparable. So the biggest difference, the driver for the lower solution recovery, is the increased cell accumulation / association in the A to B direction versus the B to A direction. So that completes our first set of case studies. The second category are related to permeability procedures. And I have two questions within this. The first question, your procedure for permeability measurement, if the recovery is less than 85%, a pre-incubation step may be employed. So this is according to our SOP. However, pre-incubation was not conducted during the method suitability validation. So please conduct the pivotal experiment for classification of test compound in the same manner as the method suitability study. Alternatively, please validate your modified method and include that as part of your method suitability validation. Right? So the emphasis here is that you cannot use a procedure for your test compound or drug substance that has not been previously validated, qualified, or shown to be suitable. So the approach we took here was to extend the method suitability to include the procedure that we had used for this specific test compound. And in order to demonstrate that pre-incubation does not impact the classification, we ran a bridging study using CACO2 monolayers from different days in culture and passage numbers. We ran this with a subset of low and high permeability model compounds from the validation, antipyrin, metoprolol, atenolol, and minoxidil. And we did this with and without a 10-minute pre-incubation, essentially to demonstrate that this does not impact the test system's ability to classify and to rank. Here are the outcomes. With pre-incubation, without pre-incubation, table on the left, very similar. Atenolol, as expected, is the lowest apparent permeability. And then you see it increase as you go from minoxidil to metoprolol to antipyrin. The graphical representation on the right pretty much shows us that there is no difference with and without pre-incubation. So the takeaways from this experiment: A, pre-incubation step does not impact the ability of the CACO2 cell monolayer to clearly discriminate a low permeability compound such as atenolol from a high permeability internal standard such as minoxidil. It also does not impact the rank order. The four model compounds behave as expected. Antipyrin, the P apparent greater than metoprolol, greater than minoxidil, greater than atenolol, both with and without a 10-minute pre-incubation. It also does not impact the measured in vitro permeability values because regardless of the pre-incubation conditions, i.e. with or without, the slope of the linear portion on which the PAP calculation is based remains unchanged. And these results were consistent across a sample of CACO2 batches and days in culture. In other words, within the established method suitability assay window. The second question within permeability procedures. Materials and methods for validation of the permeability study indicated that the plates containing the cell monolayers were shaken using a lab line instrument's titer plate shaker set at a speed of 2. However, study report states that the plates were shaken using a VWR model 3500 plate shaker set to a speed of 1. Please clarify this discrepancy. So change of equipment along the way. Method suitability had shaker at a speed of 2. We are currently using a different model shaker at a speed of 1. Please clarify. So this was again something that over the number of years that we have been running these studies, things have changed. Certain models get replaced, but it's important to make sure that the bridging is performed along the way. So the lab line instruments were replaced with a VWR model shaker, and to address the lack of impact of shaking speed, we again decided to perform a study using model compounds with different permeability values over multiple cell passages in days in culture, representative of the assay window. And the study was performed using the current shaker, the VWR, with a shaking speed setting of both 1 and a shaking speed 2. Table on the left, results are very, very similar between the two shaking speeds. Atenolol clearly lower, followed by minoxidil, metoprolol, and antipyrin. There is robust separation between the two internal standards that we used for a permeability study, minoxidil to atenolol ratio hovering around 15, regardless of the shaking conditions. And the graphical representation on the right also indicates that the data is very tight and appears to be independent of the two shaking speeds, 1 and 2. So the key takeaways, the two tested shaking speeds do not impact the ability of the cell monolayer to discriminate atenolol versus our high permeability internal standard. The rank order of the 4 model compounds was as expected. At the shaking speed conditions, be it 1 or 2, the PAPs were very similar. Essentially, the slope of the linear portion of the time course profile remains unchanged. And these results were consistent across a sample of batches and days in cultures. And all of this was within the established method suitability assay window. So my next series of case studies are on test system or method suitability. The first question here is, per the BCS guidance, you have not evaluated the expression of MRP2 in your CACO2 cell model. Please evaluate the expression of MRP2 in your model and submit the data. So this is something that we had originally not performed. Somewhere along the way, the guidance got updated to also include MRP2, and we had to extend the method suitability to include the substrate for MRP2. The substrate that we have chosen is carboxy dichlorofluorocene. We ran this several times across many batches of CACO2 monolayers, again, representing the assay window of the BCS permeability classification. Results are summarized, table on the right, and as you can see, with an efflux ratio of 10.9, we were able to demonstrate robust MRP2 functional activity. And now this is now part of our updated cocktail, QC cocktail, before cell monolayers are released for a BCS experiment. The next question here, your model compound validation reported the PAP values for 4 drugs. These are atenolol, furosemide, hydrochlorothiazide, ranitidine. As you can see, the range of values from 0.19 to 0.55. The question here being that the PAP values of the set 4 drugs are similar to the low permeability model drugs that you use, which are between 0.07 and 0.77. However, the 4 model drugs stated up top, atenolol, furosemide, hydrochlorothiazide, ranitidine, are moderate. So given these observations, there is a concern about your cell model to distinguish among low and moderate drugs. Please clarify and provide the necessary data to address this concern. And this is a good question because based on how the question is stated, it appears that we have more of a binary classification of high and not high versus the ability to separate high, moderate, and low. So the model compound data represented in this question were all from a single unidirectional experiment, single experiment with 4 replicates, other than atenolol, which the data was from system ruggedness, where we ran this many times. So when you're evaluating data from a single unidirectional experiment, there is, of course, intrinsic variability associated with the data. More on that in my next question within this case study box. So therefore, that becomes a driver, one of the main reasons why we're seeing this overlap in the permeability of moderate versus low. So a better way to evaluate whether our test system has the ability to discriminate between moderate and low is represented in my next slide. So what we did here, similar to a BCS classification study where we co-dose compounds, we co-dosed a cassette of high, moderate, low, and a 0 marker, and then ran this multiple times as a cassette. And as you can see from these outcomes, that the Keiko-12 cell model can clearly discriminate and rank compounds with different permeability values consistent with the expectations of the method suitability section of the BCS guidance. Hydrochlorothiazide, atenolol, both in the moderate bucket, are different from lisinopril and FITC, and also different from the high markers such as antipyrin and minoxidil. So running this as a cassette, co-dosed multiple times, you get a much better sense of the ability of the test system versus a single point comparison, which is what the question was based on. My last question in this section, it is noted that the permeability of drug substance is substantially differentiated in the permeability studies with and without internal standards. So unidirectional is with internal standards where you're trying to use the data for classification. Bidirectional is without internal standards to demonstrate the test system's suitability for use for BCS classification. So for example, at a concentration of 50.8 micromolar, the permeability with and without internal standards are 32.1 and 25.8. So higher with internal standard, lower without internal standard. This data indicates that the permeability may be influenced by the presence of internal standards. Please provide an explanation on this difference in PAP and its potential impact on the permeability classification of drug substance. So the concern here would be of a false positive. Is there some type of interaction between the co-dose controls, the internal standards versus the drug substance, which could be bumping up its permeability, therefore resulting in potentially a false positive? So that is the basis of the question. Our response. So we first begin with, what is the typical variability of the Keiko-2 cell monolith test system? One way to look at this is to mine the data that we have for the internal standard, which has been collected over many years. So minoxidil over a 10, 12, 14-year period has a percent CV in the ballpark of 25 to 30%. I have some more data on this. This is between 2007 and 2019. As you can see, the overall mean is about 5. This data is from a total of 128 separate BCS studies, 1431 valid determination, valid as in data was not rejected due to either poor linearity of minoxidil or failure of the co-dosed internal standard for monolayer integrity. The mean minoxidil P apparent was 5.25, median of 5.19, indicating that we likely have Gaussian distribution. And this had a percent CV in the ballpark of 25 or so percent. So with this context, and as we extend it to the question for this particular drug substance, it is not unreasonable to expect that the inherent variability for a sticky compound is expected to be probably around the same ballpark as minoxidil, likely a little higher because you also have other dynamics in play, whereas minoxidil is a clean compound which has excellent recovery and no association with cell monolayers or to the transfer. But the two values in the question only have a relative percent difference of 21.8, which is well within the expected degree of variability for this test system. So this was one way we were able to respond to this, to demonstrate to the FDA that what we're seeing is in line with the behavior of this model or test system, and that regardless, in addition, both of these values, with and without internal standard, are at least threefold higher than the co-dosed high permeability internal standard minoxidil. So we have sufficient separation, regardless of with or without internal standards, where there is no possibility that the value, the P apparent that we have calculated, can be lower than the qualified internal standard. One last note here. When we take into account the average P apparent values for drug substance from all of the experiments performed in the studies, not just the single concentration, but also the low, the mid, and the high, and put them together, A to B unidirectional with internal standards and A to B bidirectional without internal standards, the numbers are remarkably comparable, 19.4 versus 20.1. And this is how we are able to provide multiple data points to work around this issue. And finally, my last case study is on impact of excipients. So as per the guidance, when new excipients or atypically large amounts of commonly used excipients are used, then you have to document the absence of an impact on bioavailability. Your test formulation contains surfactant SLS. Please provide justification with supporting evidence that the amount of SLS used in the test formulation does not have any impact on drug absorption and also dissolution from the test product. So our focus here is on the absorption part of it. So in the test product that is the subject of this ANDA, the formulation contains 2.32% of SLS. So it is 10 milligrams of SLS in a total weight of 430. So we prepared a multi-layered response. One was trying to compare this amount of SLS to what is permissible according to the FDA inactive ingredients database. We also looked at some clinical publications where amounts of up to 50 milligram did not show a difference in bioequivalence, and that was the first layer of the response. Additionally, 10 milligram of SLS translates to, if you use the BCS volume of 250 ml, translates to 40 microgram per ml of SLS in terms of concentration. And we had data from a study performed or a study published in 2016, and the reference is cited on the right, where a cassette of model drugs, 4 class 3, acyclovir, atenolol, ganciclovir, nadolol, and 1 class 1 antipyrin were evaluated in the presence of the excipients that are listed on the left at multiple concentrations. And if you take a look at the highlighted portion, 0.04 milligram per ml, 40 microgram per ml, essentially the data is no different from the lower concentrations of 20 and 10 microgram per ml. And also this data is in line with the control data listed at the very top of the table. So we are able to provide, in addition to data from FDA inactive ingredients database, clinical publications that showed no impact up to 50 milligram amount, that here is data from experimental data indicating that SLS this concentration does not impact the permeability. One more layer to this, and this is more recent. Many of you here may have seen this representation of our in vitro dissolution absorption system previously. We call this the IDAS. IDAS combines traditional dissolution testing with a means to determine and quantify interactions with the biorelevant membrane. So basically, it's a dissolution vessel where we have two inserts, and on the inserts, we mount Keiko2 monolayers. So we're able to study dissolution and permeation together, concomitant. So we think of this as biopharmaceutics dissolution, which hopefully results in better in vivo. The apparatus has been supported by the FDA. They funded a 2-year contract, a broad agency agreement to see whether this could be used to expand BCS class 3 waivers for generic drugs to non-Q1, Q2 products. There were 2 objectives broken up into year 1 and year 2. Year 1 was, is the test system sensitive? Can it discriminate? So again, similar to the publication I had shared previously, which was the transfer format. Using the IDAS format, we also prepared a cassette of compounds, evaluated multiple excipients, 15 total at 3 concentrations. And year 2, which is now complete, we looked at formulations that had clinical BE data published and ran them using the IDAS to try and perform a retrospective analysis to establish IV-IVC with available clinical bioequivalent. So this lists all the different excipients that were chosen, 15 excipients from a variety of different categories. SLS, and these are the low excipient are the maroon bars. Again, very similar to the transfer format. There was no impact, but the benefits of this test system versus the transfer is improved dynamic range, potentially up to an order of magnitude higher versus the more traditional Keiko2 format. We are able to test 0.3 milligram per ml of SLS versus 40 microgram in the transfer, before which the transfer, so any higher in the transfer format, and we would compromise cell monolayer integrity. One more piece of evidence that at 40 microgram per ml, we should be. So summary of these results from year 1 and year 2, Keiko2 cell monolayers and IDAS are less sensitive to excipients than in transfer, hopefully more in vivo-like. This may be due to the geometry. They're vertically oriented versus horizontal, and likely more effective mixing because you can agitate the donor solution with a paddle. And most of the excipients tested had little or no effect on the permeation of class 3 drugs, suggesting that expanding biowaivers to non-Q1, Q2 formulations is a possibility. And this could have important consequences for the development and regulatory approval of generic drugs. And this is now the IDAS in practice. We are evaluating excipients, impact of excipients for a potential class 3 biowaiver. RLD versus test. The test is non-Q2, graphical representation on the left, permeability comparison of RLD and test, and statistics on the right. We're looking at receiver concentrations, AUCs, and it appears to be no different, even though the formulation is non-Q2. This data package has been submitted, undergoing review, and we're working through this currently. So with that said, that is my last case study. I thank you for your attention and look forward to participating in the panel discussion and answering any questions that you may have. Thank you.
Well, that was an excellent presentation, Sid. I really enjoyed it, and I'm sure so did everybody who was attending. Real in-depth information sharing of, working with FDA to resolve specific issues regarding Keiko2, and the IDAS example, the methodology development is very nice to see and looks promising, and I hope it yields all the promise that it's showing. We are a couple of minutes late, but I think we'll get caught up. I'm going to introduce our next speaker, Dr. Donna Volpe. It's a pleasure to introduce her. Donna is a colleague with us at FDA. She's a member of our CEDAR BCS committee, probably from the beginning of the formation of the committee. And Donna is, at least to me, and I think objectively speaking, also she is probably one of the most well-known experts in KCO2 and and cell studies, not just for permeability but also for drug metabolism and other aspects. She has published extensively on this methodology over a period of years, and I'm really happy to welcome her for her presentation, which is going to be sharing details about method suitability of KCO2 cell models for drug permeability classification. So if we can have a presentation. Thank you, Dana.
Hello, everyone. I'd like to start by thanking Jim Polli and Mehul Mehta for their kind invitation to speak at this workshop. As a disclaimer, the content of this presentation should not be interpreted to represent any agency, determination, or policy. This presentation will cover the following topics: the components of method suitability, the use of model drugs and reference standards in the permeability assays, acceptance criteria for the standards, and end with the literature examples of method suitability with KCO2 cells and their use for drug classification. Different experimental permeability values for the same drug between laboratories are the result of differences in how the assays are performed in terms of culture and transport conditions. These differences can affect cell morphology, transporter expression, and transporter characteristics. The dependability of an experimental permeability model is shown by its ability to accurately predict a drug's in vivo intestinal absorption. Standardization of the assays reduces intra-laboratory variations in permeability results and consequently improves the predictive potential of the assay. Method suitability is a generalized approach that a laboratory can utilize to standardize and validate a drug permeability model. It establishes a correlation between experimental permeability values and known human intestinal absorption. Method suitability allows for the use of different permeability assays within a laboratory, whether they involve human studies, intact animals, intestinal tissues, or epithelial cells. As described, method suitability accounts for intra and interlaboratory variability and allows for improvement in the assay technology. The combination of the three components, such as assay standardization and validation, reference standards, and acceptance criteria, enables laboratories to improve the consistency of their experimental data in an effort to predict a drug's intestinal permeability. Method suitability is comprised of three components: method development, assay suitability, and permeability classification. In method development, the first step is to establish an assay protocol in terms of cell culture and transport conditions. Next is the optimization and standardization of these assay parameters. For KCO2 cell monolayers, this may include cell source, cell passage number, culture media composition and the feeding regimen of the cells, the choice of the filter type, diameter, and its pore size, the initial seeding density, the monolayer age or the number of days the cells are grown in the monolayers, the transport buffer composition and its pH, the sampling method, and time points. Lastly, a lab sets its own acceptance criteria for different assay parameters such as the measurements of monolayer integrity, the apparent permeability values for high and low reference standards, and the efflux of a model PGP substrate. For assay suitability, once a laboratory has optimized its assay, model drugs are then used to demonstrate the rank order relationship between experimental permeability values and human intestinal absorption. Based on the result of this experiment, a high permeability internal standard is defined for the assay in that laboratory. The reference standards are then included in experiments for permeability classification with a new drug to demonstrate assay reproducibility. Molecular markers, such as zero permeability compounds, are used to establish cell monolayer integrity. The high permeability internal standard is then used to classify a new drug as being high or low permeability. For in vitro cell culture methods, it is recommended that at least 5 model drugs per group are included to establish method suitability. The selected model drug should be passively absorbed in humans and have known intestinal absorption. The model drugs represent a range of in vivo human intestinal absorption in the following groups: low permeability, that is fraction absorbed less than 50%, moderate permeability with a fraction absorbed of 50 to 84%, and high permeability with a fraction absorbed of greater than or equal to 85%. An evenly distributed data set according to absorption improves permeability prediction, since bias towards completely absorbed model compounds in the assay tend to predict high permeability well, but then lacks precision to predict low permeability drugs. As you can see in the representative graph here in the bottom right, the in vitro permeability should correlate with in vivo absorption. Reference standards are used to improve permeability experiments. High and low permeability standards are used to monitor experimental variation laboratory. A zero permeability standard is used to monitor cell monolayer integrities. An example of this includes FITS dextran, PEG 4000, Lucifer yellow, and inulin. A highly permeable internal standard, the high permeability, is with experimental permeability that is in close proximity to the high-low class boundary of the model drugs, is then selected from that group of drugs. This high permeability and standard is then used to help classify new drugs. It is important that the permeability values of the standards do not differ significantly between the different experiments, including those that were conducted to demonstrate method suitability. There are several experimental options to include these standards and the experiments with a new drug. First, you can measure the test drug together with the reference standards in the same monolayer well. The issue here is that the reference standards should not interfere with the analytical assay that is used for the test drug. And conversely, the test drug should not interfere with the analytical assay for the standards. Secondly, you can evaluate the standards in the same monolayers after the experiment with the test drug has been completed. Here, you will need to ensure that there is still monolayer integrity. And lastly, you can use additional cell monolayers that were cultured at the same time as those that were used for the test drug. For example, wells can be used on the same plate that you have evaluated the test drug. Within a laboratory, acceptance criteria are defined for the selected reference standards and measurements for the permeability assays. This includes measures of monolayer integrity that include low permeability or zero permeability molecular markers, or TEER, the trans-epithelial electric resistance, to demonstrate the presence of an intact physical barrier for drug transport. The apparent permeability of zero, high permeability, and low permeability standards can also be used. And lastly, acceptance criteria should be established for the efflux of a probe substrate compound. The acceptance criteria are used to demonstrate the functionality of the assay, and the reference standards are used to ensure reproducibility of results in the laboratory. The functional expression of efflux mechanism in a cell monolayer is demonstrated with bidirectional experiments with a probe transporter substrate. P-glycoprotein, multi-drug resistance protein 2, and breast cancer resistance protein are all important efflux transporters that are found in the human intestine. Efflux is revealed by a higher rate of a substrate's permeability in the secretory or basolateral to apical direction as compared to absorptive, which is in the apical to basolateral direction. A drug with an efflux ratio greater than or equal to 2 is considered to be an efflux transporter substrate. Rhodamine 123, digoxin, vinblastine, paclitaxel, and quinidine are often used as probe substrates for demonstrating the presence of a functional PGP transporter. Here on the right, you can see results from our laboratory where we looked at the efflux ratio of several fluoroquinolone drugs in comparison to the standard rhodamine 123. All the drugs except for ofloxacin hit efflux ratios greater than 2. In addition to efflux transporters, CACO2 cells express uptake transporters similar to those in the intestine, such as those for amino acids, peptides, monocarboxylic acids, organic cations, and organic anions. Drugs may be substrates for these transporters. When using the cell assays, it is important to maintain the same protocol from the initial method suitability experiments to those that are used to classify a drug substance permeability. Here, the reference standards are utilized for classification and reproducibility. Keep in mind that the cell-based assays are only for those drugs with passive permeability. This is shown by evaluating the test drug at several concentrations such as 0.01x, 0.1x, and 1x, the highest dose strength in 250 ml. Keep in mind that some drugs may not have sufficient aqueous solubility to evaluate at such concentrations. In that case, lower concentrations may be used. The permeability values should not differ significantly between these concentrations. If they do, the test drug may be a substrate of an uptake transporters. You should also measure the bidirectional permeability of the test drug to determine if it is subject to efflux. If its efflux ratio is greater than or equal to 2, then that test drug is likely a substrate of an efflux transporter. A test drug is classified as being highly permeable if its apparent permeability is equal to or greater than that of the high permeability internal standard. Next, I'd like to show some examples from the literature of method suitability with CACO2 cells that demonstrate their ability to classify the permeability of new drugs. The first is from our laboratory of a KECO2 cell assay in a 12-well plate format. We evaluated the model drugs using monolayers that were cultured for 18 to 22 days. Hank's ballot salt solution was used as a transport buffer with a gradient pH in the apical and basolateral chambers to mimic intestinal physiological conditions. The initial drug concentration was based on the drug product's highest dose strength in 250 mLs. This was to represent the concentration of the drug in the intestinal tract. We used rhodamine 123 as the model PGP substrate. The graph on the right shows a clear delineation between high and low permeability drugs. Here, labetalol was chosen as the high permeability internal standard as it fell at the low permeability, high permeability boundary. There was a good relationship between the permeability of the model drugs in this KECO2 cell assay with the extent of human absorption for the model drugs. In this next example, from a collaboration of laboratories in Slovenia and Germany, KECO2 cells were cultured in a 24-well plate for 21 days. The buffer was at pH 7.4 in both the apical and basolateral chambers. The initial drug concentration was based on the drug's maximal clinical dose in 250 mLs. They also used rhodamine 123 to demonstrate PGP functionality. The graph on the right again shows a clear delineation between the high permeability compounds and low permeability compounds. Here, metoprolol was at the low permeability and high permeability boundary. There was a correlation between the permeability coefficients of the tested drug substances with their human fraction absorbed. The investigator also measured the expression of uptake and efflux, transporter gene expression, finding that the expression was dependent on the monolayer age. Such things as this can lead to variability in some of the results, showing the importance of maintaining a similar protocol in the same monolayer age. In this last example from renal pharmaceuticals, KECO2 cells were cultured for 22 to 24 days in a 12-well plate. The Hank's balanced salt solution buffer was at pH 6.8 and 7.4 in the apical and basolateral chambers, respectively. The initial drug concentration was 10 micromolar for all the model drugs. Digoxin was used as the model substrate for PGP functionality. The graph on the right shows a separation of high and low permeability drugs, with minoxidil at the high and low permeability boundary. There was a rank order relationship between the permeability values in the assay and the extent of human intestinal absorption from the literature for the tested drugs. These examples demonstrate that each laboratory should determine their own high permeability internal standard based on the results with the model drugs used in the method suitability experiments. Next, let's look at two examples of how these assays were used to classify the permeability of other drugs. The graph on the left shows the use of the KECO2 assay to classify the permeability of loccaserin. Increasing concentrations of this test drug showed similar permeability values, even though the permeability at 200 micromolars looked higher. It was not significantly different between the other concentration. Metoprolol and mannitol were the high and low permeability reference standards, respectively. Here, minoxidil was the high permeability internal standard. seen here, we can see that locurcerin was classified by the investigators as being highly permeable since the PAP values at the different concentrations for this drug was greater than that of minoxidil. In the graph on the right, we used our CACO2 assay to compare the in vitro permeability of fluoroquinolone drugs. Metoprolol, atenolol, and fixed dextran were the low, high, low, and zero permeability reference standards respectively, with labetalol as the high permeability internal standard. Ciprofloxacin and lomofloxacin were classified as low permeability, whereas levofloxacin and ofloxacin were classified as high permeability. Ciprofloxacin has a bioavailability that's less than 85%, where the other drugs had bioavailabilities greater than 95%. The reason that these drugs did not match the permeability of the bioavailabilities is that these drugs are subject to varying degrees of efflux, as we saw in an earlier slide. These two examples show how method suitability can provide information on intestinal permeability of new drugs and the need to determine if those drugs are subject to active efflux. To summarize, method suitability is a generalized approach to standardize and validate permeability methods within a laboratory to investigate and classify the intestinal absorption of drugs. Method suitability for intestinal permeability assays is comprised of 3 phases, including method development, assay suitability, and permeability classification. It is important that each laboratory sets its own acceptance criteria for cell culture and transport measurements, along with the use of reference standards that are used in both the initial experiments with the model drugs and those experiments that are then used to classify a new drug's permeability. Method suitability, with its reliance on assay standardization and validation, reference standards, and acceptance criteria, helps to enhance the consistency of experimental data that is utilized to predict a new drug's intestinal permeability. Thank you for your attention.
Donna, thank you very much for that very informative talk on method suitability for CACO2 cell line, and I'm sure the audience really appreciated that and has learned a great deal about getting out this experiments successfully within their labs. So now I'm going to introduce our last speaker of this session, who's Dr. Haritha Mandula, another FDA colleague. A pleasure to welcome her. Haritha has been, she's a senior pharmaceutical quality assessor in the Division of Biopharmaceutics within the Office of New Drug Products at OPQ, Office of Product Quality within FDA. Again, she's also been a member of our CEDAR BCS committee for several years now. She's involved with the review of biopharmaceutics aspects of new and generic drug product submissions. And she's on the BCS, CEDAR BCS committee, as well as the IVC committee. Real pleasure to introduce Haritha, and she's going to be sharing with us best practices for examples. Sorry, let me rephrase that. The typical deficiencies relating to permeability assessment supporting BCS Biowaiver. So if we can have Haritha's presentation, that'd be great. Thank you, Dana.
Thank you for the introduction. The title of my talk today is Typical Deficiencies Relating to Permeability Assessment Supporting BCS Biowaiver. I would like to start with a disclaimer. The views expressed here are personal and do not represent those of the FDA. Slide summarizes OPQ's mission and emphasizes product quality. A quality product of any kind consistently meets the expectations of the user, and drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine. This slide summarizes the outline of my presentation today. I will talk briefly about BCS classification criteria, followed by the different types of permeability measurements as outlined in the FDA ICH M9 guidance. Further, I will talk about the criteria and supporting studies for high permeability classification. I will present few case studies and summarize the typical deficiencies associated with permeability measurements. The slide summarizes BCS classification criteria. Biopharmaceutical classification system is a scientific approach based on the aqueous solubility and intestinal permeability characteristics of the drug substances, resulting in 4 classes, namely class 1, which is high solubility and high permeability. Class 2 is low solubility and high permeability. Class 3 is high solubility and low permeability. And class 4 is low solubility and low permeability. The same is depicted in this graph here, where x-axis represents the volume required to dissolve the highest dose and y-axis indicates the permeability measurements. Class 1 with high solubility and high permeability is represented in the upper left corner. Class 2 with low solubility and high permeability is presented at upper right corner. Class 3 with high solubility and low permeability is at the lower left corner. And Class 4 with low solubility and low permeability is depicted at lower right corner. Coming to permeability measurements, there are several ways as outlined in the guidance for permeability measurements in favor of BCS1 classification. These include in vivo permeability measurements based on extent of absorption from human PK studies, and typically these include absolute bioavailability and mass balance studies. Human in vivo data from published literature may be acceptable. Note that peer-reviewed articles may not contain the necessary details of testing to make judgment regarding the quality of the results. In vitro approaches include permeability measurements using Keiko2 cells. The guidance acknowledges other in vitro and in situ methods, but agrees on methods with most experience. Other methods could be considered for future once they are standardized. This slide summarizes the criteria for high permeability and the supporting studies as needed under certain conditions per the guidance. A drug substance is considered highly permeable if the absolute bioavailability is greater than or equal to 85%, or greater or equal to 85% of the administered dose is recovered in urine as unchanged drug parent, or sum of parent drug and phase 1 oxidative and phase 2 conjugative metabolites. For metabolite in feces, only oxidative and conjugative metabolites are to be considered. Metabolites formed through reduction or hydrolysis should not be included, unless it is demonstrated that it is not produced prior to absorption. For unchanged drug in feces, it cannot be included for extent of absorption unless data supports biliary excretion or intestinal secretion. For in vitro assessments using validated in vitro Keiko2 permeability, the criteria is usually the results indicating high permeability from these validated systems. And the Keiko2 results should be discussed in context of available human PK data. And these are usually limited to passively absorbed compounds. Supporting studies include stability in the GI tract under certain situations. If mass balance or Keiko2 studies are used, data to support drug substance stability in the GI tract should be provided. However, Such stability data are not required if mass balance study shows greater or equal to 85% of the administered dose recovered as unchanged drug in urine. GI stability is usually documented using compendial or simulated gastric and intestinal fluids. Other relevant media with appropriate justification can be used. And drug solution is usually incubated at 37 degrees centigrade, typically 1 hour in gastric fluid and 3 hours in intestinal fluid, and eventually analyzed. Significant degradation of greater than 10% would suggest potential instability. So anything less than 10% would indicate stability in the GI tract. This slide summarizes BCS applications as received and classified as Class 1 by FDA's BCS committee. The pie chart here shows that the BCS committee reviewed 97 applications so far and classified 66 of these applications as BCS Class 1 drug products. 31 of these were not classified as BCS Class 1 drug products upon review. This bar chart on the right summarizes the different types of permeability studies among the 66 applications that were classified as BCS Class 1 drug products. As can be seen from the graph, the most common is KCO2 permeability measurements, followed by mass balance and absolute bioavailability studies. Least common are the in-situ rat intestine perfusion methods. A handful of applications did reference RLD labeling or literature and were approved. Note that some of these approved applications used more than one method of permeability assessment in support of high permeability. The first case study is that of an IND. This study is an example where the applicant demonstrated high permeability based on a mass balance study. Applicant submitted solubility, dissolution, KCO2 in vitro permeability studies, mass balance studies, and gastric stability studies. Compound A demonstrated high solubility and rapid dissolution. As can be seen from the graph here, the highest dose strength is soluble in 250 ml or less of aqueous media over the pH range of 1 to 6.8. Rapid dissolution of the drug product was also observed with greater or equal to 85% drug dissolved in 30 minutes at all the tested conditions in accordance with the guidance. The applicant submitted KCO2 studies in support of high permeability. However, permeability based on KCO2 in vitro cell culture system was moderate in comparison to model compounds. At some concentrations, the permeability was comparable to high permeability model compounds, and at other concentrations, it was comparable to moderate permeability model compounds. The data is summarized in the table below here. As can be seen here, the A to B permeability and B to A permeability was decreasing with increase in concentration, indicating saturable transport. And the B to A by A to B ratio was greater than 2 at all the concentrations tested, indicating active efflux. Cmax was dose proportional within the range of doses tested, and the applicant has also demonstrated adequate gastric stability with degradation less than 10% up to 24 hours. So based on this KCO2 measurements, the drug substance could not be classified as BCS Class 1. The applicant also submitted mass balance study. The mass balance study was designed as an open-label, randomized, single-dose crossover phase 1 study conducted in healthy subjects. Plasma and urine samples were collected pre-dose and up to 48 hours post-dose. 13 subjects were randomized, 1 subject withdrew, and 12 subjects completed the study. Greater than 85% of the administered dose was excreted in the urine. However, the variability of the measured concentrations in urine was very high based on the available data in the study report. The applicant was therefore requested to submit entire urine data sets as well as provide their explanation regarding the high variability. The applicant noted that out of the 12 subjects, only 2 individual subjects had less than 85% urine recovery. One subject was at 82% recovery and another was a clear outlier who had only 50.9% recovery. When the excretion outcomes were recalculated excluding the outlier, the mean excretion was almost 92.7% of the administered dose. This reduction showed that the variability was heavily influenced by the data from the outlier. Compound A was classified as BCS class 1 drug substance and BCS class 1 drug product based on the totality of the data submitted. Figures below here show cumulative urinary excretion with and without outlier subject. Elimination is high, greater than 85% with and without outlier. Removing outlier makes data tighter with less variability. Case study is that for an ANDA where the application is approved for high permeability based on KCO2 permeability measurements. Compound B here demonstrated high solubility in the range of pH 1 to 6.8. It also exhibited rapid dissolution with greater or equal to 85% of labeled amount dissolved in 30 minutes in multimedia pH conditions. The graph here illustrates the same, demonstrating rapid dissolution. RLD labeling indicated that absolute bioavailability of compound B when compared to IV infusion is 25% due to extensive first pass metabolism. There also existed a linear relationship across the dose range. High permeability here was supported based on in vitro permeation studies across KCO2 cell monolayers and also based on stability data in the GI tract. As can be seen from the table here, the permeability data, the compound B showed greater permeability than the high permeability model compound minoxidil at all the tested concentrations. The permeability was higher than minoxidil at all the tested concentrations with no particular trend showing lack of saturable transport, and the efflux ratio B to A to A to B was less than 2 at all the tested conditions. During initial submission, there were deficiencies pertaining to method validation, and these included missing SOPs, lack of data on zero permeability compound, missing recovery studies, which were all eventually resolved. So compound B was classified as a BCS class 1 drug substance and BCS class 1 drug product. The third case study is of an ANDA where permeability assessments were based on mass balance of RLD application. In this study, compound C was classified as BCS class 1 based on high solubility at all the tested pH conditions, as can be seen from the graph here. Rapid dissolution exhibiting greater or equal to 85% dissolved in 30 minutes in multimedia pH conditions. This is illustrated in this graph here, and mass balance of RLD application. The mass balance study in the RLD application was designed as a single dose study in 6 healthy male subjects. Mass balance following IV and oral administration of radioactive compound was evaluated. The total radioactivity was measured in blood, urine, and feces for up to 7 days. And following oral dose, 93% was recovered in urine. And finally, this was classified as a highly permeable drug substance. So some of the challenges around evaluation of mass balance in RLD applications is the fewer subjects involved and higher variability. Here, all the 6 subjects demonstrated greater than 93%. However, sometimes there are cases where the data is inconclusive. 3 of the subjects exhibit less than 85, and then 3 subjects would exhibit greater than 85. And in cases like this, usually the permeability assessments need additional support from another kind of measurement. Later in the year, few other ANDA applications did not pass 90% criteria for FED-BE studies. This was further investigated, and the data indicated excipient food interactions. Basically, the high proportion of microcrystalline cellulose and swelling of microcrystalline cellulose matrix under fed conditions lowered the rate of absorption, and eventually waiver of fasting BE study was granted, but fed BE study was required. The fourth case study is of an ANDA where permeability assessments were based on in-situ rat intestine perfusion method. Here, compound D demonstrated high solubility in aqueous medium over pH range of 1 to 6.8. And multimedia dissolution data demonstrated rapid dissolution with greater or equal to 85% dissolved in 30 minutes. And the same is illustrated in the graphs on the right side, solubility data and dissolution data. Data from CACO2 studies in the NDA showed moderate permeability. However, in vivo human pharmacokinetic information and scientific literature indicated that the bioavailability for this compound is high. In this ANDA, the applicant conducted in vitro permeability using in situ rat intestine perfusion method. And as can be seen from the permeability data in the table below, that the permeability measured at all different concentrations was high than the high internal standard metoprolol measured here. And hence this was classified as a high permeability compound. Later investigations on the CACO2 assessments led to the finding that the compound being cytotoxic was causing loss of integrity, and hence CACO2 is not an appropriate model for the permeability assessment in this case. This was also supported by the stability studies in the gastrointestinal tract, and percent degradation was less than 10%, indicating stability of the compound in the gastrointestinal tract. Slides 15 through 17 summarize the typical deficiencies associated with different methods of permeability assessments. This slide here summarizes the common deficiencies associated with absolute bioavailability and mass balance studies. So these deficiencies are usually due to lack of study reports, validation reports, and other procedural documents. Sometimes there is unexplained high variability in the measured urine concentrations, and the applicants are requested to provide justification or additional documentation. The applicant is also sometimes requested to provide complete data sets along with calculations. There are also missing GI stability data to support, especially when less than 85% of the unchanged parent drug is excreted in urine. Coming to typical deficiencies associated with CACO2 studies, these can be categorized into 2 categories: deficiencies associated with pre-study model compound validation, and deficiencies associated with the pivotal or the actual permeability study. Typical deficiencies with the pre-study model compound validation include missing standard operating procedures, missing validation data with model compounds, missing calibration curve, and quality control data, including linearity, slope, precision, and accuracy for all analytical runs used in the permeability study, and sometimes missing data on zero permeability model drug. So these are the common ones. There are usually more than this, but I have just listed the most common deficiencies. Coming to the deficiencies associated with permeability study, usually lack of non-specific binding permeability study, the procedures describing insurance of pH 7.4 during the course of the study, and data to demonstrate stability of stock solutions, high recovery rates of internal standards, and differences in recoveries between apical to basolateral and basolateral to apical directions. Permeability measurements influenced by presence of internal standards, difference in permeability measurements of internal standards between validation and pivotal studies. Deficiencies or concerns with literature-based or labeling-based permeability assessments usually are lack of necessary testing details for peer-reviewed journals or unreliable RLD label data, usually large variability, fewer subjects, etc. And some of the deficiencies associated with instability in GI tract assessments are missing procedural SOPs, like study conduct, preparation of buffers, etc. Reference is made to FDA ICH M9 Biopharmaceutics Classification System Guidance, which is finalized on May 2021. I would like to acknowledge the following individuals and divisions and offices: Kimberly Rains, my branch chief, also my division director, and also chair of the BCS committee at FDA, Utpal Munshi, another chair for the BCS committee at FDA, Mehul Mehta, previous chair and also one of the organizers of this workshop. He's also a member of the FDA BCS committee, Division of Biopharmaceutics, Office of Bioequivalence, and Office of Clinical Pharmacology. I would like to acknowledge the following individuals, divisions, and officers for all their support. Kimberly Rains, who is my branch chief, Paul So, a division director and chair of the BCS committee, Utpal Munshi, also chair of FDA BCS committee, Mehul Mehta, who was a previous chair of the BCS committee and who is a current member of the BCS committee and also one of the organizers of this workshop, FDA BCS committee, Division of Biopharmaceutics, Office of Bioequivalence, and Office of Clinical Pharmacology. Thank you for your time and attention.
Well, thank you, Haritha, so much for that excellent presentation. And I'm sure the audience would have found a lot of useful information there, particularly the typical deficiencies seen in the submissions we receive, and that would help them put together a better package the first time around and reduce the cycle time. So I think we are just a couple of minutes behind. And Jim, I think now we're going to get into the panel discussion, and I should be handing it over to you.
Okay, Mehul.
Thanks, Jim.
Right.
Yeah.
All right, thank you. Thanks to all of our speakers. And so, as Mehul said, we're pretty much on time, so we're very fortunate to have the next 45 minutes or so to be able to take your questions. I encourage you to type questions into the chat. So at this time, we would invite all speakers who spoke this morning to join the panel by turning your video on. We also have a couple additional panelists we're very fortunate to have with us. I'll introduce those three individuals now. Jan Welink from the Medicines Evaluation Board. Jan has worked since 1997 as a senior clinical assessor at the Dutch Medicines Evaluation Board. He was chair of the Pharmacokinetic Working Party of EMA until recently, and he remains an expert member of the group. His specialist areas are in the area of bioavailability, bioequivalence, and BCS. Currently, Jan is involved in ICH-M10 on bioanalytical method validation as deputy topic leader, and ICH-M13 on bioequivalence as regulatory chair. Also, Shereeni is joining us from Health Canada. Shereeni is a senior assessment officer to Health Canada, where she evaluates biopharmaceutics information provided in support of approval of innovative and generic drug products. Her expertise includes pharmacokinetics, comparative bioavailability and modeling and simulation for biopharmaceutics applications. She served as Health Canada's topic leader on ICH-M9. Also, John Gordon, who's giving a perspective of WHO, although works at Health Canada. For 24 years, John has been an assessment officer at Health Canada. For a vast majority of that time, he's been involved in international activities, particularly in the areas of bioequivalence and BCS Biowaivers, through his involvement in the WHO pre-qualification team medicines. He's been directly involved in the development of the bioequivalence and BCS-based guidelines published by WHO and served as WHO representative to M9. So thank you all for joining us this morning. Thank you again for the talks. Let's see. We do have our first question here. Again, I realize we have several hundred people. If you can type questions into the chat, either to everyone or send it to Paul So privately if you so wish. Let me just read the first one here. I am hopeful that the FDA and other agencies can still consider the rat intestinal perfusion model. That model has been successful for many years. I assume that was not included in M9 guidance is because companies were using the rat perfusion less, but not because the rat mucin was shown to be invalid. Let's see. I think that question was for regulatory agencies, but maybe we could just ask Sid as a starter. Sid, I think you presented some perfusion data.
Sister Jim, not in this presentation, but the same thing crossed my mind. It is related in a sense because I saw Haritha share her case study on case study 4, I believe, was on in-situ perfusion, and it also crossed my mind. Will that persist going forward? Because such compounds or drug substances will exist or persist, which are cytotoxic to cell monolayers. So even though I don't have my perspective, I would still hope that if it is sufficiently validated, why would it be dissimilar to case study 4?
Okay.
Jim, I can comment also.
Okay, great.
Just with regard to the M9, you should take into account where it's coming from, which countries are involved or which regions. I think FDA was one of the very few regions which has implemented in vitro data as pivotal data to prove permeability and absorption. For the other region, this is a new area. They don't have experience with that. And even with KECO2 cell data, there was some resistance to use it. So I can only indicate, count your blessings on this moment that we accept in vitro data, and then it's in this case the KECO2 cell data, for proof of permeability. Because there was already some resistance to include it also in the M9. What the future will bring, I cannot predict, of course, but of course, knowledge will evolve, regulatory agency will evolve, and new opportunities may be possible, and also with regard to the M9 to include more in vitro, let's say in vitro studies to prove permeability. Thank you.
And yeah, this is Mehul, if I could just add to that, and as Jan explained, and then as people have shown through data, that was Haritha's slides too. And when we were at the ICH, that was, as we have it, FDA has had maximum amount of examples and data on KECO2. And so when sharing that and going over that data, that's what everybody in the M9 group felt comfortable. So if you remember in the FDA guidance, initially we had those other methods also suggested as possible alternatives. But now this is where, as Jan was saying, the maximum level of comfort and data is with KCO2. So that's why we adopted that. And moving forward, there's scope for expansion with, again, with sufficient data.
If I could just add as well to that.
Okay, Shereeni.
So at Health Canada, our initial guidance on BCS-based biowaivers in 2014 did consider other in vitro models to be supportive information. But at that time, our guidance also was, it relied on the extent of absorption for designation of permeability class. As you've heard from Jan as well as Mehul, with M9, it was really the comfort level with the amount of experience across regulatory agencies that dictated our use of only KCO2 cell assays for this. So I do hope that moving forward with maybe more experience around these models, that we'll be able to move forward on that too.
Okay.
So Jim, just raising my hand here for practice. That's one of the things I actually advocated is maybe next time we meet, we can think about what it takes to make people comfortable and actually put criteria by which you could validate new methods in as an alternative for trying to decide at that point in time, does everybody feel comfortable with a current method? If you can decide on the methodology, then you don't need to update as new methods come along, as long as you're confident what you have for the type of data needed to allow such a method to be used and the performance it needs to have.
Jim, if I could just follow on that just a little bit, I agree. WHO, of course, is not a regulatory agency in themselves, but they provide guidance to be used by other regulatory agencies. So the possibility of using, in vivo or in situ intestinal perfusion using animal models has been in WHO guidance since 2006, but the sense I've gotten from people from various regulatory agencies sort of outside the ICH core is just the uncertainty and not knowing how to deal with that sort of data. So I think that what Jack suggested is that making better information available so that there's a better understanding of what this data could be used for and how to assess that data, I think will go a long way to making it more accepted. I think having KCO2 as a standardized approach in M9 because it will be accepted, across everyone using the M9 model, will increase the comfort level more quickly because you'll see it more commonly, and other regulatory agencies will see it more commonly, and they'll become more familiar with it, and then you go from there to the other models that Jack and colleagues think would be just as good as KCO2.
So, Jim, if I can, sorry for the interruption, but I mean, not outside of this point, but, and I think Jan and John and Shereeni have already started doing that, but if that's okay, and if, you accept my suggestion, if Jan, John, and Shereeni can share some further perspective from their respective agencies or organizations about BCS, just to orient the audience further about utilization of BCS in their respective organizations. That'd be my suggestion, and if that's possible, that'd be wonderful. Thank you.
Yeah, that'd be great. Sorry for jumping into the questions, Jan. Is there anything else? Thanks for talking about getting to the first question. Just backing up a little bit. Anything else anything that you can relate to us in your observations over many years with M9?
Yes, thank you for this also, because I think it's also important with regard to the EMA, what the regulatory environment is. We come from a long way, and that's not only because the guidance was not in place, but also we have to face with 27 member states. That makes it rather difficult also for applicants, but not only for applicants, also for regulators. Already before the 2010 guidance, there was already an opportunity for more or less biowaiver applications, but the guidance and the criteria were not very well embedded in the guideline. Of course, the 2010 guideline came in force, however, still it missed clear guidance, clear criteria, what cut-offs were, for instance, what kind of similarity excipients were allowed or not allowed. And this triggers a lot of discussions, but also a lot of problems, because every member state can object to a decision of another member state. So we were happy that the M9 came into force. The M9 at least has some good and clear criteria which should be fulfilled for a biowaiver application. So that helps most of the member states, of course. Also, what I want to address is the biowaiver principles is waiving of a bioequivalence study. It's not lowering the criteria for proof of bioequivalence. That should not be misunderstood. So discussions of, okay, we can go forward further with maybe moderate permeable drugs, or we can extend the guidance to drugs which are not highly soluble at a certain pH. Those questions, I find this at this moment not in place to ask. That is also not the case if you have an in vivo bioequivalence study. You know the criteria, 80 to 125%, for instance, is a hard criteria. Even if you are 1% outside it, maybe not clinical relevant, but your application will not be accepted based on that. You should look at, in this respect, also the M9 guideline. The criteria are there, and if you fulfill them, you will get a biowaiver. Thank you.
Thanks, Jan. Shereeni?
Thank you, Jim. Health Canada implemented the harmonized M9 guidance in August of last year, so it hasn't really been very long. Prior to that, we did have a guidance from Health Canada. The draft was in 2012 and was finalized in 2014. That guidance, as I mentioned before, relied on the extent of absorption for the designation of permeability class, and data based on in vitro methods was considered to be supportive evidence, and that would also include Keiko-2 cell assays. With the implementation of M9, it meant a change back to the original BCS classification for us, and the acceptance of in vitro methods using Keiko-2 cell assays as pivotal evidence for permeability. Since about 2019, when M9 was endorsed, we've received more submissions with BCS-based biowaiver requests or justifications, and also more inquiries regarding BCS-based justifications. To date, we've had about, sorry, not to date, but between 2019 and now, we've had about 20 submissions, 16 of which were from generic drug products and 4 were for innovator products. We're also beginning to see more Keiko-2 cell data in support of high permeability, but it's still very early days yet, and I'm hoping that with more time we'll see more companies adopting this, and therefore likely an increase in submissions with this. Deficiencies we've noted to date relate to inadequate validation of the methods, and that's very similar to what Haritha had described in her presentation, We see missing model compound data often, and also incomplete documentation. The presentations this morning have been very interesting and informative, and I expect that discussion at forums such as these will promote the use of the biowaiver approach in the future and probably lead to increasing numbers. Thank you.
Thanks, Shereeni. John Gordon is also from Health Canada, but I know he spent quite a bit of time working on behalf of WHO.
That's right. Not from Health Canada today. Yeah, so I can speak from two WHO perspectives. First of all, again, it's WHO in general, not a regulatory agency, but providing guidance and policy that can be used by regulatory agencies. And to go along with that guidance as much as possible, sort of a training, workshop training is provided to familiarize people from regulatory agencies, again, outside the ICH core, generally speaking, with these approaches, be it BE or BCS-based biowaivers. And as I suggested the last time I was speaking, the sense from the regulatory agency was the uncertainty about how to deal with these in vitro methods. And I get a clear impression in the training in the last couple of years is that they're very happy to be able to focus on, for now, CAECO2 approaches, because then they can concentrate their efforts on building, capacity to evaluate those things, those studies appropriately. And presentations like we saw today would be very much appreciated, I think, by these people from these agencies, because this is the information they need to better understand the pitfalls and the details that they need to understand. So I think the CAECO2 is it's been a very positive. M9 has been very positive in establishing CAECO2 as a standard, and there's information in there that they can refer to, and I think you'll see a much greater acceptance of these types of studies, internationally, and I'm very happy about that. But other models I think can follow, but I think time is needed to establish the core capacity from these countries. The other perspective I can speak from, of course, is the only regulatory aspect of WHO on this perspective is the pre-qualification of medicines program, and that program works with the defined set of drug substances and drug products. So they're not like a regulatory agency in that a company can't just submit the product that they're working on. It's a defined list, and for that reason, the program hasn't seen a lot of in vitro data to support a biowaiver, because quite often, you have products like levofloxacin, ofloxacin, and so on, where the permeability or the extent of absorption is well defined, and it's well known. So, from that perspective, there hasn't been a lot of in vitro data presented, but I suspect as the list of products that can be submitted to pre-qualification grows, and the familiarity with CAECO2 approaches grows, that we will see more information, more studies being submitted to the program.
Okay, thank you, John. Back to some questions. If you have any questions, please type those in the chat. We have one for Jack Cook. This has to do with timing, I think, of experiments. So Jack, you mentioned BCS classification permeability assay is done in preclinical stage and in phase 2. Can you elaborate on the value to do the phase 2 permeability assay according to ICH M9 standards? Or when choosing not to do this, to more elaborate on ICH M9 standards?
So yeah, because of the limited capacity, and I think there are references to using the mass balance study, we actually use what we gathered in preclinical studies to look at the likelihood, look at the likelihood of it being classified as a highly permeable compound. And if the likelihood is not high, we may not use the methodology to look at the mass balance. Those are pretty powerful studies. They can answer other questions, so it wouldn't rule it out entirely, but it wouldn't have in its favor of being one of the candidates for the study to run that quarter, that we would be able to solidify the highly permeable ranking. And so that's how we decide. A few are filtered out early in development if we're really lucky to have a compound that's eliminated unchanged in the urine. That's it. I think the first one we submitted was on pregabalin, and that was indeed the case where we had over 90% of the drug excreted unchanged. So hopefully that answers the question.
Okay. Anyone else want to comment about what to do when, with a view towards having BCS biowaivers in your pocket or needing to apply one?
I just can comment from a regulatory point of view. If you look, and then I'm looking as an innovator dossier, the amount of data is of course different than the amount of data a generic company has and what is in it in a generic application. So the totality of data which has to be taken into assessment is totally different. And if, for instance, the M9 is not fully applied to, for instance, to support BCS, there are more data than only these BCS data. At least there could be more data, and that will be taken into account. So we will not say, oh, you did not fully apply, for instance, the M9 to support the BCS classification. There could be a different assessment and a different evaluation. But still, finally, we are convinced that if we make the decision, that we make the right decision what kind of BCS classification it should be. Thank you.
So Jan, just to clarify, you were giving me a Christmas present there saying that you're open to a totality of evidence approach when the evidence exists.
Sorry to interrupt. Yes, but we do that always. However, I already indicated an innovator company has more data, mostly more data. Yeah. They did a lot of studies which are confidential for a generic company. And generic companies are leaning on what is publicly available, for instance, or they have to do their own studies. And that makes a totally different package, of course.
Any other comments about what to do when?
I will just add one more, and that is I'm an advocate of trying to do it and finding out the answer before you need it, rather than deciding I've got one more bioequivalent study to do. I need to hurry and do the assessment to see if I can apply for a waiver. And that's why I actually advocate trying to get it done around the time of phase 2 before you have to do a switch in formulation that requires some type of documentation of bioequivalence.
And yeah, I would just add a bit more on the lines of what Jan already said, and also supporting what Jack said. But again, at the innovator stage, as Jan was saying, there is a lot of data available with the innovator company, and as Haritha presented an example where totality of data was used. There was KECO2 data, there was In-Vivo data, and then so KECO2 was not convincing, but In-Vivo was convincing, and then more data for In-Vivo results was asked for. We have examples where I think Jack, you had a bullet point on that, but that's an actual example where KECO2 was low permeability, but In-Vivo there's very good data that there's like gangbusters type extent of absorption with good In-Vivo data. So that can happen, and then so that's the pleasure or privilege or for innovator company to have all that data, and then if that's brought together well, and as Jack, you were saying about timing, then that decision or that viewpoint can be established in a timely manner, so that you don't need to worry about hurrying up for a BE study. So, again, as Jan said, on the new drug side, we are big, and I think our BCS committee also, we look at all available data and looking at the generic side, as Jan was saying, but there's limited information available. But then there's all these additional tools available where generic company can benefit from BCS-based approach. And I published some data a little while ago, and Haritha had more update on that. But we have, or even on the innovator side, a lot of BCS based biowaivers considered and granted, but a lot more on generic side, a lot more. So and that's why luckily we have that database that we've been able to share. So some broad perspectives in timing. Thanks.
If I can add just one more thing, I appreciate the support and the statement that totality of evidence and the other can be utilized. I will say that a challenge for some innovators are that as we apply to agencies and health authorities throughout the world, all it would take is one to be more conservative, where you would have to do a bioequivalence study in humans for that agency, and that's what we default to. And so if one of the places we're going to file says we're not going to accept this other data because it's, for instance, not listed in M9, that means that we won't pursue that.
Just one thing to add before I forget it. Of course, it is important when you want to apply a waiver, for instance. Depends which kind of formulation has been used in clinical trials. Are there pivotal clinical trials or not? Or have those formulations used a lot in the clinical studies or not? And it becomes more critical, for instance, if you have a formulation changed at the end of all clinical trials, even with phase 3 studies. Then, of course, uncertainties in BCS classification become more critical. Thank you.
You.
Do you
Question? Sorry, Sid?
I just kind of picking up on the conversation that we're having. This question comes to us a lot, and I think the audience may benefit from hearing the regulatory viewpoint on this. Many applicants look at the FDA's product-specific guidance, and this is from the ANDA perspective, and many times the guidance may say, perform a BE study, fed, fasted, but there is now a deeper understanding because the FDA has been applying biowaivers for many years now. There is a deeper understanding that this is a scientific framework. So therefore, if your product meets the criteria as defined, you can move forward with a request for a biowaiver, even if the product-specific guidance today specifically only talks about a human PK study. On the European side, again, my understanding is more recently there are also these product-specific guidances which have been published for a few different drug products. So my question was, do we also look at that similarly? Some products will say, you cannot move forward with a biowaiver request because it has been determined that solubility cannot be met, or this is unstable under certain pH conditions. Those statements, again, to a reader may appear unequivocal. So the FDA product specific more as a guidance. Should the applicants also be reading EMA the same as in more of a guidance, a starting point, or if something states this is unstable, solubility has shown not to be met, is that more of a period that you cannot really attempt to provide data which may supplement or overcome that?
Yes, thank you. The product-specific guidance are more or less or more focused on generic applications. Of course, those criteria in the product-specific guidelines also apply to innovator products, especially if they have already been marketed and they will change their product at that moment. But what I mentioned before, there is some gray area. If it's during development, at what time you want to apply a waiver for a formulation change. How pivotal are those formulations, and can you make a risk-based assessment that, okay, there could be some uncertainties, but you have information with the final product which has been used, for instance, in phase 3 studies. So this becomes more or less a little bit a gray area. Thank you.
Okay, let's get to a question from the audience. This is for Katja and Katja. The question deals with efflux ratios and in vivo versus in vitro and that sort of thing. So you showed that there was no in vivo evidence of PGP effect of desloratadine through grapefruit studies and linear dose and linear absorption. However, it was said that desloratadine was nominated as a PGP substrate on in vitro data. The CACO2 data shows indeed efflux, however, this efflux ratio is 1.7, below the general threshold of 2.0, considering a compound to be an efflux substrate. I guess here's the question. Do you expect the compound could be accepted for BCS Biowaiver if the efflux ratio was greater than 2? And there was no in vivo evidence that it was actually transported in vivo.
Thank you for this question. We got this data from the literature that it is a PGP substrate, but this is only proven through in vitro data. So in vitro, we know that we have overexpression of PGP. But there was also a useful indication that grapefruit juice, which is known to inhibit PGP, has no impact on linearity of pharmacokinetics. So obtaining in vitro, in our experiments, efflux ratio of 1.7 is not a surprising effect, because we know we have a PGP overexpression, and it is not over the limit of 2. So we can say that it doesn't have so profound effect. So everything was in accordance. It was not, contradictory data.
All right, another question. Thank you. Another question. This is for Haritha, at the FDA. One example you showed involved rat intestinal perfusion, slide 14. If the drug was cytotoxic to CACO2 cells, why was it considered not toxic to rat intestinal epithelial cells during intestinal perfusion?
Thank you for the question. I would expect it to be cytotoxic even to the rat perfusion model, but maybe, this is just a speculation. Maybe at the concentrations tested, the in vitro system was more susceptible to the cytotoxicity, and at the levels tested, rat intestine model was better. And also it was not solely based on, the assessments were not solely based. There was also data at the time of NDA, like in vivo data, suggesting that it was high permeability. However, when the innovator tried to use the CACO2, they were getting results that it's like a medium permeability. So I think as previously discussed, based on the totality of the data, it was convincing enough that this could be a high permeability compound. So others can chime in too, but I don't think, if it's cytotoxic to CACO2, it wouldn't be to the rat perfusion model. It would behave almost similar.
Okay. The last two questions had to do with apparently different performance in different in vitro methods, as well as sort of differences between in vitro and in vivo, for example, efflux. Does anyone want to say anything at the top of their mind about these topics, which I think we often think about?
So Jim, one comment I'd make is going back to the desloratadine question. if I'm remembering this correctly, this would fall under the category of non-drowsy antihistamines. So you run this using overexpressed MDCK with MDR, you'll probably come up with a robust efflux ratio. And if you use a cell line that is more representative of the intestinal phenotype, then it's the data that Katya just talked about, which is less than 2. So I think it's providing context. And you could still have situations where in vitro results in a 2.2 or a 2.5 or 2.73. I don't think, at least from our perspective, it does not mean that you're not passively transported drug substance. There could be sufficient evidence based on human PK and so on that what you have is essentially an artifact of the expression levels, which is generally exaggerated or overstated in vitro versus the human situation.
Okay.
Yes, but also when you have typical PGP substrates, you don't get efflux 2, you get 10 and more.
That's right.
Higher.
Yeah.
Yeah, I think I agree with Sid. I'm not sure what word to use. The example that comes to mind is the prototypical PGP substrate, digoxin, right?
Yeah.
If you go to some of the guidance documents about drug interactions, including transporter-mediated drug interactions, digoxin is a prototypical substrate for PGP. But it doesn't necessarily appear that PGP modulates its permeability. So, what does that mean in the context of this group here, where we're trying to understand what tools can we use to assess biopharmaceutic risk? So I think there's just some underdevelopment of the science, whether that's, overexpression in a cell line, or maybe it's expressing properly, just in vivo. It just doesn't seem to make as much difference as you might guess just from that particular tool.
So Jim, with that particular one, it was a byproduct. It's a product-specific sometimes interaction. Some of the newer digoxin products have a bioavailability of almost 100%, and therefore inhibiting CACO won't increase it over 100%. Yet some of the older products had a much lower bioequivalence. You may find that that's more reflective in the solubility and dissolution of the formulation rather than the permeability.
Yes, and it also depends on where the formulation is releasing your digoxin.
Yeah.
If it's in the upper part of intestine, there is lower expression of PGP than in the bottom, in the ileum.
Okay. So let's see, we've got about 8 more minutes before so-called lunch break. Let's see. This is a CACO2 methodology question. So maybe Donna, but anyone can take it. Would regulators accept CACO2 cell line grown on the expedited media, for example, 4-day confluency, as referenced this morning, versus the standard 21-day system?
I'm trying to think back to see if we had any examples of using an accelerated system. But I think what is more important, whatever system you're using, that you do show method suitability, that you show that your assay system can go and classify a drug as high permeability or low permeability, including your model drugs, and using your reference standards with the assay. So I don't see why that would not be accepted as long as there's the data to back it up in your validation studies.
Okay, another question here, though it may seem still to be too soon to see the full impact of M9. Did regulators see any change in types of submissions versus pre-M9? For example, M9 is more restrictive for BCS class 3 in the EU than the previous guideline. Was there any impact on how many class 3 submissions are made? Not sure if this is any data or if it could be disclosed yet, just in case trends could be already seen. So I think the question has to do with, pre-M9 versus M9, which is coming along 2 years, right? Any particular trends about the impact of M9 that can be observed?
Yeah, I'll start with us, FDA, and then and others can share their experience today. But we have been pleasantly surprised. We have already seen post M9 its utilization. FDA has received BCS class 3 based biowaiver applications, several of those. And if I recall correctly, we have approved at least some of them. I don't have a clear number about how many were received and how many were approved, but that was a very clear trend we saw post M9. So there has been this discussion and concern regarding reverse engineering and figuring out exactly the composition of the RLD to the extent that you meet the Q1 Q2 limit set for class 3, but it seems to be doable, and we are starting to see applications showing that. So at least I felt that was definitely a positive development post M9. And others, please share their thoughts. Thanks.
I can drop in. It was mentioned already. It seems that the question indicated that pre-M9 EMEA assessment with regard to BCS class 3 drugs was less strict than post M9. However, I think that has a little bit different origin, because there were no clear criteria what the difference could be. The guidance already indicated that there should be comparability in excipients, and for critical excipients they should be similar. However, it was not explained what is similar. For instance, what are similar amounts? That was not explained. And then you get an issue of assessment, and an issue of assessment between regulators between different countries. I remember cases where, for instance, the SUPAC principles were applied to support allowable differences. However, SUPAC is not applicable, of course, in the EU region. Still, it could be a tool at that time, a tool to assess BCS-based biowaivers and certainly BCS3 drugs. So the M9 gives a more clear guidance, and already mentioned, if you follow it, you will have a certainty that there will be no major issues in the EC region. Thank you.
Just commenting from Health Canada standpoint, we've actually seen maybe increased numbers since M9 was endorsed. However, we only implemented M9 in August, so since then there hasn't been that much time. And I would agree that we've seen more submissions, but that's related more to the fact that generic companies are now able to use KECO2 cell assays as pivotal evidence. Prior to M9, we also had slightly more restrictive criteria, I would say, for the comparison of excipients for a waiver for BCS class 3 drug substances. So compared to that, now we do have seen a couple of pre-submission requests with formulation differences that would meet what we're requiring per M9. So there are encouraging signs that we will be seeing an increase in that as we move along.
Okay, thank you. So we have just a few more minutes, so we'll take maybe just one more question. Sometimes the cell line KECO2 is sensitive to drug concentration being tested in the relevant range. For example, that the drug may be cytotoxic in sufficient concentrations that could cause cell death. Any suggestions as to how to address this when it occurs? Katja, Sid, or Donna?
I think it's important that we've seen cases where concentration may be cytotoxic. You just might have to go use lower concentrations of the drug in your KECO2 assay system.
Right, and to add to that, Jim, I would say consider maybe reducing the incubation duration. So those are two levers that one could potentially use.
Yes, but in worst case, if you're limited by analytical limit of detection and quantification, you could use animal tissue because it's more of the tissue and it has some, let's say, better resistance to cytotoxic than monolayer of cells. But I don't know if the regulators would accept this kind of scientific justification for the cytotoxicity.
In my experience, I think it's probably true that rat perfusion is less susceptible to toxicity than a cell culture line, for example, like a monolayer.
Mm-hmm.
Okay, since it's not quite 11:30, just one last question. In your experience, which regulatory health authority has been demanding on bioequivalence rather than biowaiver? And what are their major concerns for rejecting biowaivers? Okay.
I'm not answering.
Okay. Should Ravi answer then?
I cannot reveal what Board of Health authorities we face this kind of situation, but it does occur, and it has occurred even for BCS class 1 drug as late as even last year.
Mm.
Okay.
I can comment from a regulatory perspective. It has to do with, now we have M9, for instance, in force. The criteria are clear also for BCS1. And if you don't fulfill the difference in amount of critical excipients, then you cannot apply for a biowaiver. So, and that is the issue for instance, in Europe.
Mm.
In the instance, sorry to just interrupt, in the instance that I mentioned, it is the same formulation composition, 2 different manufacturing sites for a BCS class 1 drug, and we were denied biowaiver. And they both are approved in their respective markets. And it's the same source of API, same composition of excipients, same source of excipients, still not acceptable.
So, Ravi, I don't know if this is the case, but I will say that it's pretty apparent before M9 was released, there were a number of countries who were not open to BCS based waiver comments, and so of course you would receive pushback when we would try to go to have them consider that, and that's why I put trumpets on the slide that I do again want to applaud the efforts, because it does establish a baseline that I'm hopeful that most, if not every country will, and regulatory authority will accept.
Yeah, I'm also hoping the same, but we are already sensing some challenges in some of our current compounds where we are trying to get a biowaiver for a lower dose strength when we have done a bioequivalent study for the higher dose strength. And it's a common blend, so it's a proportional composition. We still cannot get a biowaiver. So these are some real challenges that we face in real world.
Okay.
Well, might be pointing out that there has been some major movement. Japan is a significant market for some companies, and Japan has gone from not accepting biowaivers at all to adopting ICH-M9. So I think that's a big step that should be acknowledged.
Yeah, we are very optimistic that the M9 is going to change the landscape.
Yeah, hopefully your experience reflects just that pre-change, Ravi.
Mm-hmm.
Okay, I see that we're a little over, so locally here, Eastern Time, it's 12:30, and we'll reconvene in an hour. But please don't come back to this WebEx. No, I'm sorry. Please do come back to this WebEx. What we'll do is we'll have one lecture by Jennifer Dressman on future permeability testing. And in the meantime, you can start thinking about what questions you may have for the breakout sessions that you will participate in at 2 and 3 o'clock. Okay, so see you back at this same URL, WebEx URL, in an hour. Have a great lunch or early dinner or early breakfast if you're on the West Coast.
Thanks, Jim. Thanks, everybody.
Thank you.
Thank you.